Perfect. So, welcome and great to have you on board to today's PAR Foundation webinar series. Today's agenda is packed. We're in World AMR Awareness Week, and I have a good lineup today with me. 4 speakers who you soon will meet. My name is Cecilia Dahlström. I am the Secretary General at PAR Foundation, and with me I have Gabriel Oke, who is our Communication Manager. He will monitor your questions. But before I hand over the word to our speakers, let me just briefly tell you about PAR. Our vision is really to be part of eradicating the spread of antibiotic resistance. We have been working on that since 2017, and to do so, we support research and educational initiatives that are working in the area of prevention of antibiotic resistance. Since the start, we have funded 25 projects across the globe. I'm glad to say that we have our first Latin American project on board this year. And we have built a community of 1,000 passionate individuals working with these topics over the years. And since the start, we have distributed over 16 million of Swedish crowns in these projects. If I talk briefly about this week then, we have a full program. There is still an opportunity to join us tomorrow. We will do a Twitter space tomorrow at 11 o'clock Central European Time. At that meeting, we will release micro grants to individuals who have been part of our World Antimicrobial Awareness Week challenge. So join us tomorrow at 11 o'clock if you can. But without further ado, let's jump into today's agenda then. Our first speaker is sitting in Sweden as well, Sepehr. I'm going to hand over to you to do the very brief introduction of yourself, and then I hand over for you to talk about your research.
Thank you, Cecilia.
So do you want to tell me briefly just what you're doing at?
Absolutely. I will also explain more in the presentation. So my name is Sepehr Shakeri Yekta, and I'm the researcher at the Department of Thematic Studies, Environmental Change at Linköping University. So here we work quite a lot with environmental challenges related to contamination and resource use, and in the presentation I intend to explain a little more how AMR comes into the picture or into the work that we are doing.
Fantastic. Thank you.
Thank you.
I will stop sharing and I'll let you take over.
So I would appreciate if you confirm that you can see the presentation.
Yep, absolutely.
Great.
We see your screen now.
Thank you so much, Cecilia. So, and also before I start, I would also like to thank PAR Foundation for the opportunity to be able to present our work on mitigating the environmental spread of AMR through waste and wastewater treatment. And so we are a group of researchers from with different backgrounds and competences. We try to tackle this information. So in this group, we have researchers from the Department of Thematic Studies, Environmental Change at Linköping University, the place that I am, which we collaborate with other researchers at the Department of Medical and Clinical Science at Linköping University. But also it is a collaboration, a close collaboration with the Department of Medical Microbiology in Federal University of Rio de Janeiro. So my aim with this presentation is simply present the project that we have initiated and the approach that we have for mitigating the environmental spread of antimicrobial resistance. So just a brief information about how we work here at Linköping University. So as I mentioned, so we have two challenge areas to consider. One is related to the resource use and the other related to a spread of contaminations in the environment. And as you can see, the roots of contaminations from both urban activities, agricultural activities, but not the least industrial activities. And our ambition simply with the research is to try to redirect the material flow in a way that we actually look into waste and wastewater as resources rather than as problems. So we are working with developing technologies to simply use resources for material and energy recovery in the social system, in the urban systems, industrial systems, but also related to the agricultures. So, and AMR is a very key aspect in this, because when we are dealing with the waste, so we also need to handle this aspect as part of the other contamination or contaminants that we are dealing with. So we have created a vision which is to try to remove clinically relevant AMR genes along with the material or nutrients recovery and energy recovery from organic waste. So we have initiated the project. It's funded by CNPq, which is a funding agency in Brazil, and the Swedish Research Council. So we have focused mainly on technologies for managing sewage, food waste, and manure, which has been shown that can contribute to environmental spread of AMR if not handled properly. So if we just look into the dissemination routes of AMR, so we can see that the urban activities, for example, through unmanaged sewage disposal and runoffs, are important routes of a spread of AMR into the natural systems. And not the least, when we have even the management system in place for waste handling, like the sewage treatment plants or organic waste management plants, handling plants. So there is also very little studies on how the downstream flows from these treatment facilities will contribute to the AMR spread. And to the right, we also have agricultural activities, very common practice using, for example, manure or residues from the agricultural activities as fertilizers in the and that will on its turn can contribute to AMR dissemination. So our goal simply is to target these flows by developing and placing treatment strategies that particularly optimized for removal of the antimicrobial resistance. So we have set three main objectives. First, we would like to characterize AMR genetic trace in different organic waste, but also we would like to assess the fate of NMR genes across the existing waste treatment infrastructure. And by this knowledge of the genetic traits of AMR, which is in our waste flow, so we would like to develop treatment solutions. So we started our work in Rio de Janeiro, so where we have we think to address the unmanaged sewage disposal system. So here in the top right corner of the picture, you will see the map of the Rio de Janeiro, and the blue line is a Carioca River, which is a very characteristic river for the city. And a problem related to that is that the sewage is disposed directly to the river, which will then end up to the Flamingo Bay, which contribute, and measurements have shown that the bay has high levels of antimicrobial resistance. So in this work, for example, we have selected a number of points from the mouth, from the source to the mouth of the river. So we have collected the samples, and we try to simply characterize the AMR and actually how both the spatial and temporal variations of the AMR genes based on the in the river and connected to the city activities. So and to build on, and in this case we have this unmanaged sewage disposal system that we would like to know about, but we also know that there are many management systems in place. So in another parallel study, we also went around and launched a campaign to look into the genetic traits of AMR in different types of waste in waste management systems. So we have went to waste treatment systems like sewage sludge, food waste handling, or manure handling plants in Sweden, Denmark, and also somehow later in Norway. So, and our goal with this study, which is ongoing, is to map the AMR genes in different treatment plants. So, and this information will simply help us to kind of move forward. So it will be a background to move forward to develop technologies for the removal purposes. And the technology that we would very much focus on is related to its anaerobic digestion, as you can see in this picture. So we have in Linköping what we call a Biogas Solutions Research Center, which is a research center focusing on the services that the anaerobic digestion and biogas production systems can provide to the society. So just to kind of try to motivate why we are focusing on this specific technology. So this is anaerobic technology, is a biological technology that we are working with the mixed microbial cultures, as you can see in this picture. And this microbial community, it's a complex community capable of handling complex wastes. So we need a community that can deal with different characteristics in different types of waste. But the outcome is that this process actually allows us to convert waste to products. It can be energy carriers like methane, which can be used as fuel, like hydrogen as well. We can also use the technology to produce chemicals that have applications in chemical industry, but also, not the least, we can use this technology to produce biofertilizers. So this technology is somehow in place, but what happens with the AMR in this technology is a very kind of new field of research. We know very little about that when we use this technology for treatment of the waste. So what will be the fate of NMR? So for example, one study that we have launched, so we try to map actually the changes in the AMR genes characteristics from the simply bin to the land when we have the food waste management. So this is an example of industry which takes in the food waste and process it in different steps, and in the end produces biogas, but also delivers biofertilizers to the farmers. So then that will be very important for us to see how these processes actually helps to either mitigate or not the further spread of antimicrobial resistance when the outflow will be used as biofertilizers. We also, as I mentioned, this is a collaborative project with Federal University of Rio de Janeiro. So we are in parallel working with developing monitoring method, which is currently missing in our field, so that we would like to develop methods that we can do when we are optimizing and we are running the technology. We can also monitor the AMR genes. So for that, for example, we have established a parallel research lab in the University of Rio de Janeiro. So now we have 2 labs that we can address this question related to the challenges which are actual for the local. So just to give you a brief overlook, so our approach is to we try to target clinically relevant antimicrobial resistance genes. So we apply both cultivation-based methods combined with the PCR and sequencing, but we also complement it with metagenomics to identify antimicrobial resistant genes, but also mobile genetic elements. So then we use this information to develop reactors and technical processes, and then we try to optimize the operation. And hopefully these will give guidelines to the full-scale actors that running full-scale treatment plans to try to optimize their operation towards the removal of the antimicrobial resistance. And we also follow up their performance, and that will also help us to modify our optimization techniques. So if you're interested in collaboration, of course, we will be very glad to have more knowledgeable people on board for this purpose. So please don't hesitate to contact us. And by that, I thank you for your attention.
Thank you so much. I will ask Gabriel. You are monitoring the questions on the back end. Do you have any questions for Sepehr?
Can you hear me?
Yes, we do.
Okay, so I can't see. So there's no question for now. But if we get questions later, maybe we'll just share later. But for now, there's no question from the audience at the moment.
Okay. Thank you so much.
Thank you.
And thank you. Then I will hand over to our second speaker today, Pontus. Can you tell me a few words on yourself, and then I will hand over to you?
Yeah, thank you so much. So my name is Pontus Nordenfelt. I'm an associate professor at Lund University in Sweden. And we work on different ways to create antibody treatments. And I'll come back into that, why that will be relevant for antimicrobial resistance. But we're working a lot with different methods and approaches to antibodies.
Excellent. I'll stop sharing and I'll let you share instead.
Yeah, I'll share my screen. Let's see here. If this works. Does it look okay?
Absolutely. Thank you.
Great.
Take it away.
Okay, I'll just move this out of the way. Okay. So, yes. So I will tell you a bit about our project that we just started. We already have some interesting data. And it's about new effective antibodies against Pseudomonas aeruginosa. So, first, I'll just briefly introduce Pseudomonas aeruginosa, which is a multi-drug resistant opportunistic pathogen listed as a priority 1 by WHO due to the high resistance and prevalence of the bacterium. And it's one of the leading causes of infections in immunocompromised individuals, such as those with chronic pulmonary disease, or COPD, or KOL in Swedish, cystic fibrosis, severe burns, sepsis, and cancer patients. And since the antibiotic resistance is highly prevalent, and it's also these bacteria have a way of getting resistant as well, new treatment options are in urgent need. So I mentioned before that we'll talk a bit about antibodies, so I need to introduce that a bit. So this is a representation of an antibody. It's typically described as a Y, and they have 3 domains, 2 identical ones, so two Fab domains and 1 FC domain. And the Fabs are the ones that recognize the targets, in this case a bacterium, and they can then give different effects just by binding to a protein or structure. They can block further binding of that a bacterium, for instance, or they can neutralize adhesion properties or other functions related to the target that they are binding to. But then we also have the FC part, and that is essentially a flag to the immune system. And that has a tremendous amount of different effects, such as phagocytosis, where the antibodies trigger immune cells to eat the target. We can have the activation of a complement system, which is another system that can mediate protective effects, inducing inflammation, and other effects. I won't go through them all, but there's a long list of different potential important effects. And there are different classes and subclasses of antibodies, and they are capable of inducing different types of these FC-mediated functions. But for most approved drugs, the Fab-mediated effects have been focused partly because it's much easier to control, and you don't want to have to worry about non-specific inflammation effects. But what we are thinking about is both to use the Fab-mediated effects, but also see if we can use more of the FC-mediated effects. So I want to just briefly mention the methodology that we're using. Let me just shift one thing here so I can show with the pointer. So we have a pipeline to generate antipathogen antibodies. So we start with the pathogen, in this case, Pseudomonas aeruginosa. We have convalescent patients, that is those that have been recently infected but are recovering. And from these, we purify B cells, and B cells are the cells that produce antibodies. And from the B cells, we want to extract genetic information that describes essentially the recipe for the antibodies. Once we have that genetic information, we can build the structure. So an antibodies have 2 parts, so a light and heavy chain. And then we can produce, in this case, human IgG, which is one type of antibodies, the most common one. And then we have a whole pipeline of profiling. So we look at different biophysical aspects and functional aspects of these antibodies. And we have used this successfully against anti-streptococci, where we discovered a protective antibody that bound with both of its 2 Fab parts that recognized a protein here. And it was only that type of antibody that could protect against infection. So we're trying to develop that further and see if we can come up with a new treatment for streptococci, which are also a big problem. So back to the Power Foundation project. So what we're doing here is that we're taking 2 aims. One aim is to generate new antibodies, and the other is to engineer them to become better. So we have B cells from cystic fibrosis patients and healthy donors as well. And the reason why we pick out cystic fibrosis patients is that these patients, they're in constant contact with these bacteria, but they can't clear them properly. But since they're in constant contact, they will probably evolve better antibodies. Problem is that due to the disorder, the mucus problems and such in the lungs, the antibodies can't get to the right place. But they should be a good target for generating good antibodies. We have clinical isolates of Pseudomonas bacteria. And then, as I described before, we'll bait B cells with these bacteria, extract the genetic information, and recreate the antibodies from each B cell, and then engineer them for improved function. So the team that we have is an experienced team, 3 people within the lab. So Sebastian Wright, an MD PhD, that did his PhD on antibody discovery. We have an experienced lab manager working a long time with antibody genetics and function. And the same thing with our senior scientist Berit Olofsson. And this is a collaboration with a clinician, Lisa Pohlman, who is in close contact with cystic fibrosis patients on a daily basis. So what's the progress on the project? So we have collected B cells from 5 healthy donors and 10 cystic fibrosis patients and 4 clinical Pseudomonas isolates. And here I present some data for that. We did some characterization just to see what is the overall reactivity. So in this axis, we have the 4 different strains just numbered here. Then we have our healthy patients or donors. And then here we have the 10 cystic fibrosis patients. And this is a scale, so yellow means a strong signal and blue means a weak signal. So what we can see here is that most of the healthy donors actually have already antibodies that can recognize Pseudomonas. So these bacteria are bacteria we constantly come in contact with as well, also as healthy individuals. But what we can see is that what seems to be correct in our hypothesis is that these cystic fibrosis patients, they have a much stronger reactivity to these bacteria. So that's a good or promising start. And then we picked one of these individuals to start with, a healthy donor. And this is just to show that there was a binding from all the patients and donors except one to this clinical isolate. And we generated 6 antibodies from this donor. And in 5 of the cases, we could show here by, this is called gel electrophoresis, where we have we can separate protein chains by size, and antibodies consist of 2 chains, so a light and a heavy chain. And we can see we have 5 intact antibodies being produced. So then we wanted to test them to see if they were functioning. And what we could see is that these monoclonal antibodies, that means that there are only one type of antibody, and they bound strongly to 2 of the isolates. And we had, the antibodies that wasn't produced didn't bind, and these are positive controls that we have. But so that's promising. So we got 2 antibodies or 5 antibodies that all could bind to 2 different types of Pseudomonas bacteria. And what you can also see here, there's might not be easy to see on your screen, but there's a weak binding also up here, and I'll come back to that. So if you just look more detail about this data, we can see the intensity levels that actually these monoclonal antibodies, 4 of them bind quite strongly compared to the positive controls. So they seem to be have high affinity likely, which is good. And these are 2 different clinical isolates looking quite similar. And as I mentioned, there was one that was looked to be weak binding. So if we look at the data here, we can see that these 2 antibodies appear to be binding, but weakly, to this strain. And if we go back to looking at the initial patient and donor data, we can see here that this donor should have binding to these 2, but we also actually see this weak binding here. So this antibody that we found actually reflected what we saw in the overall antibody population of these patients, which also gives a good indication that our approach will probably be quite promising for when generating antibodies from these patients, which is what we are currently doing. So since the project just started, essentially a month or so ago, we don't have much more data to report, but in summary, I can say that we have collected B cells from cystic fibrosis patients and healthy donors. We have collected clinical isolates of Pseudomonas aeruginosa, which is important to be able to test and bait the B cells. And we have baited B cells with these bacteria and generated antibodies from these. And so far, we have generated 5 monoclonal antibodies, and they all show reactivity to Pseudomonas bacteria. And what lies in the close future is that we will generate more antibodies, and functional testing will follow. And with that, I also want to acknowledge to people in the lab again. So Martin, Berit, and Sebastian have done most of the work, but there are other people in the lab also involved. And funding, which is for most of the general of the lab, from Vetenskapsrådet and Wallenbergstiftelsen, but also now from the PAR Foundation, which we are super grateful for. So if people are interested, please feel free to contact me. You can find our lab webpage here, and on X, we have a possibility to reach out as well. So with that, I'm happy to take any questions.
Thank you, Pontus. It's exciting to see the progress. I have one question for you. What is your dream outcome with the project?
The dream outcome is to find one or more monoclonals that seem to be binding to many of these clinical isolates, and that then seem to also function in, for instance, animal models to be able to potentially become a new treatment. So I think that's the dream within the time frame of this project. And if that works out, then we have a very interesting basis for taking further, perhaps getting closer to clinical use.
Thank you. I hand over to you, Gabriel. There are some more questions.
Okay, thank you very much, Pontus. That was nice to really hear from you. So we have a question for you, and then we also have a question for the first speaker. But for you first, since you are still on the screen. So how does the administration of the antibiotics at the patient treatment work? Do you get that?
Yep. Normally there are some antibody treatments available in general, not for Pseudomonas, and they are often administered through intravenous injection. And that seems to be safe, especially if it's a human base for the antibodies. But what we're thinking here, if we're talking about cystic fibrosis patients, is that the antibodies probably have problem reaching because of the defect mucus. So we were thinking of administering them through a nasal spray, perhaps, or some way. But that will be a part where we are not experts, so we would have to rely on others to figure that out.
Okay, then don't they mess up the immune system of the patient?
No, I mean, this is one of the most commonly used drugs in the clinic. It's called IVIG, so it's intravenous immunoglobulins. So it's essentially a mix of antibodies from thousands of patients, and that's what you can give if you don't really know what to do, and seems to be quite helpful. And also that was the basis before we had vaccines and antibiotics. Actually, it was quite common to give plasma treatments to give antibodies in the early 1900s. So it seems to be safe.
Okay, good. And then do you put in mind other Pseudomonas strains?
Yes, this is just to find our first antibodies. Once we have promising targets, we will test these against many strains of different variety.
Okay, thank you very much. That'll be all. And these questions are actually from Adela Mekrova. She's also a grantee for this year. So those the questions you answered are from Adela. All right. So to the first presenter, if you're still on the call, how do you monitor and track the resistant gene in the waste?
For now, we are using ordinary methods that we have based on either culture-based approach, but also DNA extraction and quantification by both target, but also we are doing metagenomics and sequencing as well. So, but the whole idea for now is simply to see what is the input and what is the output, or what happens during the process. And so we operate the system over a longer time, and then we follow up the dynamics and changes that happens in the process. We are also hoping to move forward to have more targeted monitoring approach, but at this point is the ordinary culture-based or based on DNA extraction, and then conventional methods.
Okay, thank you very much, Sepehr. So, Cecilia, we can go ahead with the third speaker. Thank you.
Thank you all. I will hand over to our third speaker today, Luís. Nice to have you on board. Will you give us the elevator pitch on who you are, and then I hand over?
Okay, thanks, Cecilia. My name is Luís Melo. I'm an assistant researcher at Center of Biological Engineering at Minho University in Portugal, in the north region of Portugal. And since the beginning of my PhD, I'm interested in working with phages, and both from the fundamental part and also now to a more applied research. And that's what I'm going to talk you today.
Fantastic. I'll stop sharing. I'll hand over to you. Looks good.
Okay, thank you very much.
Fantastic.
So my talk will be more with not so many results and more as a description of what is being done on phage therapy, more on the Western Europe and United States. So you all know that antimicrobial resistance is a huge problem worldwide, and that leads to, amongst other problems, to a huge amount of different infections, and several of them are related with the formation of biofilms. And this is a kind of division of device-related infections and tissue-related infections that are usually caused by different AMR pathogens. So, as you also probably know, in 2017, the World Health Organization prioritized different pathogens for suggesting both academia and pharma industries to joint knowledge in develop new antimicrobials, and that's where we are now. From our part, we are working with phages, which are viruses that are specific for bacteria and the most predominant entities in our planet. And as they are viruses that specifically infect bacteria, they are the bacterial natural enemies. And as this talk is more focusing on control of bacteria, they possess different features which makes them attractive for therapy. So they are very specific for a pathogen, being consequently harmless to the natural flora in opposition to antibiotics. They only replicate at the site of infection, so they only replicate where the bacteria is present, so they will only replicate at the site of infection. In comparison with antibiotics, for example, the selection of phages for therapy is quite fast and relatively expensive to produce in comparison. And for now, all the reports that all in vivo studies and also some clinical trials, none of them report serious adverse effects on the application of phages for therapy. So I think we are now in an era where we can try to apply phages more commonly in hospitals. And the main point is that they are effective against antibiotic-resistant bacteria because their mechanism of action is not related with the acquisition of antibiotic-resistant genes. So they can work in the same way in both sensitive and resistant bacteria. Regarding a little bit of phage history, they were discovered in 1915 and 17 in both France and UK, and they were kind of used in the First World War, but after the discovery of antibiotics, they were not so used in the Western world, although they were used from there till now in the former Soviet Union. For in the last years, when antimicrobial resistance emerged, we here in Europe and also in the United States, Canada, we rediscovered them, and we are trying to know more about them, because when they were used in the First World War, no genomes were known. Some phages are not proper for therapy, so we have to know more about their biology in order to develop them as better tools to combat AMR pathogens. So, as other viruses, phages can just have the lytic cycle, but some of them can have the lysogenic cycle, and for therapy purposes, only the ones that have exclusively the lytic cycle are recommended. To develop a good phage for therapy, we need to usually do some steps of characterization since their isolation, and observe them in microscopy, determine the host range, determine their pharmacokinetics and pharmacodynamics, study their stability, study intensively their genome. It's quite important in order to say that this phage is proper for therapy. Regarding isolation, we are used to say that phages are isolated usually where the bacteria, where their host is present. So I can give you an example that the majority of the phages that we isolate are from sewage, because on sewage the majority of the bacteria that we are interested are also there. But of course we can use some more specific fountain or source for isolating phage. For example, if I want a phage for Staphylococcus epidermidis, I can use some skin swabs. If I want a phage for E. coli, I can use feces. So we can use different sources and more specific according to the presence of bacteria. Then we always observe them on the microscope. We have, I'd say, nice images. They are quite different, and with some biology knowledge, we can understand their behavior also initially based on their morphological aspects. Host range is also something very important to characterize a phage, because I told you that the phage can be specific for usually at the bacterial species level, but rarely a phage is able to kill all isolates for that specific species. So usually what we have different phages for a specific organism, and we try to find the one that is more appropriate to the one that kills most that bacteria. And you have here examples of a phage, different phages against Pseudomonas aeruginosa, where you see these red squares. Those red squares means that this phage is not active against this particular strain. So yeah, that's what we are dealing with. But usually, as phages are quite, in some species, easy to isolate, we can find phages for the majority of the pathogens that we are looking for. Genomic analysis is also something really crucial nowadays. This is an example of a phage that we isolated against Helicobacter pylori, where the phage has an integrase. So integrase is the enzyme responsible for the integration of the phage genome in the bacterial chromosome. So this phage is a lysogenic phage, of course not the most recommended for therapy. So if this phage integrates in the bacterial genome, it will not kill the bacteria by entering on the lytic cycle. So it's not really appropriate for therapy. We can define different clusters. This case is on Staphylococcus phages. by studying their genome, what they have in common, what they don't have, compare with other phages, and knowing their most relevant proteins, what they have in common, what they have in different. And the more we know about their genome, the more proteins we can say that they have a real function rather than say that they are hypothetical. We are more comfortable in assuming that this phage is more appropriate for therapy, because several years ago, 60 to 80% of the phage genes were hypothetical proteins. So, of course, it was called dark matter of phage genomes. This dark matter is, with increasing knowledge, is being decreased, but of course there are still several hypothetical proteins in some phages, but at least the ones that are being used for therapy are showing really brilliant results. Just for those of you that will be more curious about phages, it's probable that on 99 point something% of publications you will find phages belonging to these three different families: Podoviridae, Myoviridae, and Sepoviridae. They have different properties on their tails, but what I want to highlight is that taxonomy changed last year. Those Myo, Podo, and Sepoviridae families were abolished from the International Committee on Taxonomy of Viruses. So now we have different classes, subfamilies, genera, species. So it increased a lot. It's now the classification and taxonomy is more based on genomic properties of phages rather than morphological. So if you see now in the publications, probably these numbers will be less used in the more recent papers. For therapy, the quite standards that we have, of course, what we want is to have like phage banks as we have here, where we have dozens, hundreds of different phages, and when the phage bank receive the sample of the pathogen isolation, it's probable that in maximum 24 hours they say this phage is the more adequate for this pathogen, and it will be then develop a treatment prescribed by a doctor. The treatment will start, and it will be monitored. Of course, if it can also happen during treatment to have the emergence of resistant phenotypes to phage. Of course, in this case, we need to apply other phage from the collection. Also is here something called phage training, that is kind of an adaptation to the phage to the host, and this is in vitro with culture media, so it's quite fast, and in one day we can have a proper phage for those resistant pathogens. So this is a kind of way that the phage therapy is working. Also on Belgium, it was probably the country where phage therapy really re-emerged in Europe. They have an application of phages called the magistral phage. We are not talking about phage cocktails to apply to a population. We are talking about phages to being applied in a kind of personalized medicine, where we have studying the pathogen that is infecting the patient, and they also have a phage bank that when they receive the sample, they produce the suitable phage for that patient. That production the phage is produced according to a monograph. That monograph is on the pharmacopy, and it has to be prescribed by a doctor, and that's how phages are being applied. Of course, depending on the type of application, phages can be mixed with some carriers, for example, hydrogels for topical application of phages, or on some sprays for inhalation if the infection is in the respiratory tract. The number of cases of application of phages is really increasing. We saw a lot of different application of phages being published in the last, I would say, 2 years. Application of phages in real patients with really nice results, and actually in Belgium they submit a preprint that of the first 100 cases where they applied phage therapy in patients. In our group, what we do, we are involved in these networks of isolating phages and providing phages to some countries that are applying phages. In Portugal, we have the first patients that reach our lab and asked for some help. It's been difficult to contact and having approval from some doctors, but the first steps of phage therapy in Portugal occurred in the last year, and I think in the next 6 months we will have something also here published on the first cases that we applied phage therapy in Portugal. We are collaborating with the Belgian team, and we have here also, not on real patients, but on an animal. We have just a case of a rabbit where he has a nasal infection, very difficult to breathe, and we were able to isolate a phage applied to this rabbit. And I will not pass the video because I'm running out of time, but just to know that
I'm actually going to interrupt you because of time.
Yes.
Maybe you jump to your conclusion.
Yeah,
We will be able to share this afterwards.
This is my last slide. So
Okay.
I'm just saying that we cured the rabbit with phages that we isolated in our lab, and of course I want to acknowledge all the team involved here in University of Minho and also in Belgium. And thanks for your attention. And if you are curious and want to share more things, I have the contact here on my slides.
Thank you, Luís. We will be monitoring and following the work that you do. So thanks again. I will jump straight over to you, Johannes.
Yes, hi. So just briefly introducing myself, I work at the Department of Chemistry and Molecular Biology at the University of Gothenburg. I'm a biophysicist, and I mostly work with bacterial cell envelopes and try to figure out the molecular details of these. Now, I'll just start sharing here, I guess.
Fantastic. Yes, please.
And yeah, what I'll be talking about today is a bit the organization of the bacterial membrane, especially bacterial outer membrane, and how we can use this, well, maybe as a potential target for antimicrobials more in the future.
Thank you. Take it away.
Before I get into it, just briefly acknowledging the various funding sources involved in this work, and a lot of people involved in this work. Particularly want to point out Asad, who was a master's student working with me last year. Then Noah and Selin, who worked at my old group in Basel a lot. And also Kate, who is working at Uppsala University. All right, let's get into it. Starting a bit earlier, it is May 1692. We're in the mid of the 9-year war, and it's, well, basically France versus everybody else in Europe. And what we particularly want to look at is a battle for the city of Namur in Belgium. And at the time, the French military engineer, Sébastien Le Presse de Bourbon, was responsible for laying siege to the city of Namur, which at the time was in Dutch hands. Now, Mr. Vauban had a huge advantage when laying siege to the city, and that was he knew exactly how the fortification of the city worked. So the year before, he had secretly snuck around the city of Namur and watched the castle walls, watched the walls of the citadel, and he knew exactly where to strike to basically attack the city. And that gave them a huge advantage, which resulted in Namur basically being taken within less than 4 weeks. Now, why on earth would I be telling you this? Is it because in historical conflicts, roughly 90% of the victims actually fell to infectious diseases and not in battle? Because they actually did. But the reason I'm actually telling you this is for the obvious similarity between the city of Namur and the problem that Sébastien Le Prince de Vauban was facing, and our main enemy in antimicrobial resistance, and in particular pathogens of the gram-negative escape group. Because, well, basically, if we look at it, all the antimicrobial targets that we can target with antibiotics are located within a very strong wall inside of these bacteria. So be it the metabolism that we can target, be it protein synthesis that we can target with antibiotics, be it DNA or RNA replication, everything that we have that's targeting these cellular structures first needs to go across the wall of these cells. And in that sense, we actually have quite a disadvantage because unlike Mr. Vauban, we don't really know a lot about how this protective structure that surrounds the cell is actually organized. We know little. We know that it's made of 2 membranes in case of gram-negative bacteria. There's an inner membrane, and that's basically very similar to all our cellular membranes that we know of. And there's an outer membrane. And the outer membrane is quite an obscure structure. It's a very asymmetric lipid bilayer. It has regular phospholipids on the inside, seen here. And it has lipopolysaccharides, which is extended glycolipid on the outside. And in this membrane, you have embedded a bunch of membrane proteins, which are mostly responsible for letting things through. And this membrane basically forms a very strong sieve, which only very selectively lets things through. And a lot of the antibiotics that we have that work against gram-positive bacteria, which do not have this outer membrane, basically do not work against gram-negative bacteria, mostly because things never get through. So small molecules like this penicillin, for example, here, can actually get through these proteins into the bacterial cell and then be effective. But a lot of larger antibiotics never even get into that. And, well, as it turns out, we don't really have any experimental means to look at bacterial membranes so far, simply because they're so complex, we can't really rebuild them in the lab to work with them. And it's also been so far quite tricky to isolate them. And that's basically where I come in. And the 2 things I want to talk a bit about more today is, well, 2 ways how we can get into the castle. So, well, what options do we have? On the one hand, we can try and sneak through the doors. In that sense, that would be the membrane proteins. And the other option we have is basically just tear down the wall. And as it turns out, we also have a few means of doing that, even though we don't know exactly how they work by now. So what I'm mostly working with are bacterial outer membrane vesicles. And when we look at electron microscopy images of bacteria, we always see these small protrusions on the surface. And, well, if you don't know what it is, you don't care and just think this is a regular EM image of gram-negative bacteria. If you know what they are, then you see they always have, and you see them on all pictures if you know what to look for, there always are these small protrusions on the surface. And those are outer membrane vesicles. Now, what these basically are, they're vesicles that are expelled from the bacteria and they form from the outer membrane. So the outer membrane bulges outwards, and then these vesicles fill with components from the bacterial periplasm, and then they're released into the surrounding environment. And there's still a lot of debate on why bacteria actually are doing this. There's a lot of cellular functions they might be involved in, starting from quorum sensing to extracellular digestion of macromolecules, cell-cell communication, the delivery of toxins to host cells, and so on. I don't really want to get too deep into why cells are producing these. The good thing is, and what we are basically using them for mostly, is they give an ideal microsystem of just isolated bacterial outer membrane and periplasm inside. So we get a very clean sample that we can use for biophysical experiments, which gives us direct experimental access to this bacterial outer membrane. And we maintain the asymmetric structure of the membrane. We maintain the native protein content of the composition of this membrane, and we basically maintain the periplasmic content as it is in the intact bacteria, only without the intact bacteria. And what we can do is we can actually modify these vesicles quite nicely. We can control the protein composition inside of the vesicles, so we can basically enrich these vesicles with certain proteins we're interested in from the bacterial periplasm. And we can also modify the membrane composition of these vesicles and basically change the protein composition as well as the lipid composition of these membrane vesicles simply by changing the actual bacteria where we collect these vesicles from. And the way this is actually working is mostly based on a bacterial strain that was developed in Basel in the 90s, where all of the major outer membrane proteins of E. coli are deleted. So normally, as you can see on this STS Phage gel here, which just gives you an isolation of the bacterial membrane, the outer membrane is dominated by these 4 major porins here: LAMB, OMP-F, OMP-C, and OMP-A. And in Basel, they created a deletion strain called BL21D3OMP8, where all of these are deleted. And this one has an almost empty outer membrane. And we can basically just replace these 4 membrane proteins in this strain with all other membrane proteins that we're interested in. Another strain that was developed in Italy is also a deletion strain where LAMB, OMP-F, and OMP-C are deleted, and the only proteins left is OMP-A. Is OMP-F, and OMP-A is actually deleted, LAMB is deleted. It's also a very nice strain to work with because this one produces a lot of vesicles, and this one we're mostly using for producing periplasmic proteins and enriching these vesicles in periplasmic proteins. And then if you look at the vesicles that are modified on an STS Phage cell, you can see in case of a membrane protein, you get one very strong band, and this would be the protein that we overproduced. The same for the periplasmic components. We get one very strong band of the periplasmic protein that we're interested in. In this case, it's a periplasmic beta-lactamase. And we also do get all the proteins that are in the membrane, in this case, mostly OMP-F. So this would be this large band here. Now, what can we use this for? Well, we can actually use this quite nicely in an OMV-based enzymatic assay. So we can enrich these vesicles with a beta-lactamase, so with a resistance gene for penicillin resistance, which enzymatically degrades penicillins. And we can then add a substrate, a chromogenic substrate, and so this is called Centa. And this substrate will diffuse across the membrane into the vesicle and react with the enzyme inside of the vesicle. And when this does this, we get a shift in absorbance from this molecule. In case of the undigested molecule, this absorbs at roughly 350 nanometers. And if it encounters beta-lactamase and is cleaved, this absorbance shifts to 405 nanometers. So we can quite nicely monitor how the resistance gene inside of the vesicle interacts with this substrate molecule. And if we monitor this over time, we see in case of the vesicle, a slow increase in absorbance at 405 nanometers. So the substrate gets across the membrane, gets into the vesicle, reacts with the beta-lactamase, and the signal increases over time until all the substrate is digested. And this is when this actually gets quite interesting, because if we compare this to the purified enzyme, so just the enzyme in solution without the vesicle around it, the reaction for the purified enzyme happens a lot faster. Now, why is that? Well, one thing we do is something called in situ NMR spectroscopy, where we look at the structure of proteins inside of this native environment of the vesicle. And when we compare the structure of the protein inside of the vesicle and the structure of the protein free in solution without the vesicle, we realize that they're basically identical. So there's no structural difference really implied by the native environment, and we can assume that the protein isn't really affected either in solution or inside of the vesicle. So there's not much difference there. The other thing we can do is actually look a bit into Michaelis Menten enzyme kinetics and see how the enzyme inside of the vesicle reacts with the substrate and compare this to the free enzyme. And what we see there is that the reaction in case of the free enzyme is actually a lot faster. So you note here that the axis for the orange graph are different from the axis of the blue path on the right side. And the reaction is a lot faster, but the way the enzyme and the substrate interact are actually quite similar. So we also get the same, almost identical Km value for both of the reactions. So we can also conclude from that the enzyme isn't really affected and reacts with the substrate in the same way. So why do we get this difference in reactivity? Well, what happens if we destroy the membrane? So again, coming back to this time course here, in orange, the reaction that we get inside of the vesicles. And these other three graphs here are the purified enzyme, so just the free enzyme in solution. And when we destroy the membrane, either with a detergent, something like Triton X100, or with an antibiotic that targets and disrupts the outer membrane, like polymyxin B, then we actually get back the original or the unaltered signal. So what we can really conclude from that is that the transmembrane diffusion plays a huge role. And of course, we can then use this assay and see how molecules that are supposed to inhibit the beta-lactamase actually react with this system. And we can use basically the only inhibitor that is commercially available for and used in clinical practice for beta-lactamases of the metallo-beta-lactamase kind. It's called captopril. And if we add this one, the reaction is completely abolished. So we can conclude from that also the captopril has the ability to cross the membrane, get into the vesicle, and inhibit the enzyme inside of the vesicle. The same thing happens when we add EDTA, which is also an inhibitor for the beta-lactamase. If we use others like this magnolol, for example, here, which is also a published inhibitor for metallo-beta-lactamases, we see that absolutely nothing happens. And from that, we can conclude this magnolol actually, even though it has the ability to inhibit the free enzyme, it never manages to get across the membrane. And well, we can do this for a lot of experimental beta-lactamase inhibitors, and without going into much detail here, we do see huge differences. So some of them actually get across the membrane quite well, whereas others seem to not ever get across the membrane, which means even though they have the ability to inhibit the beta-lactamase and be quite useful potentially, they never reach their target inside of the cell. And well, we're not really entirely sure yet what's the reason for this, because all of these molecules are roughly of the same size. It seems that solubility plays a huge role there. But this is still under investigation, and we hope to use the system a lot more in the future to really figure out what it is that drives molecules to have the ability to get across the membrane, and what inhibits other molecules from getting across the membrane. So overall, we can basically use these vesicles in a quite nice enzymatic assay. We have a substrate, gets across the membrane, reacts with the enzyme inside of the vesicle, and we can do this with all kinds of beta-lactamases by now. And we can use this to see whether potential inhibitors for this resistance gene actually manage to get across the membrane, whether they get across the proteins of the membrane, and we can nicely select compounds which never even manage to get across the membrane. And hopefully using this, figure out a way in the future on how to more efficiently get molecules across bacterial outer membranes. Now, the other thing, and this is what I want to talk about for the rest of this presentation, that's membrane disruptive compounds. Because you've seen before that when we add either detergents or antibiotics of the polymyxin group, which disrupt the membrane, then the reaction is a lot faster. And we can also use vesicles quite nicely in this context to see how the membrane reacts to these disruptive compounds. And if we compare, for example, the action of two polymyxins, polymyxin B and polymyxin E, on the bacterial outer membrane, and compare this to just the function of a detergent like Triton, then we see in case of the polymyxin, there's actually a very abrupt change. So with decreasing concentration of the polymyxin at high concentration, the membrane is disrupted. We get the full activity of the beta-lactamase, and then there's a very sharp transition. If we get to lower concentrations, at some point, there's basically only the slow reaction, and there's not really a sharp transition between the two. So in this case here, when we get the high values, the membrane is disrupted, and then suddenly, if we drop below a certain concentration, the disruption is not happening. And this is quite different from the way detergents happen, or detergents act on the membrane, where we get more of this gradual decrease in reactivity when we decrease the detergent concentration. Now, why is that? And what's actually quite interesting is that this sharp decrease in signal that we measure here coincides directly with the minimal inhibitory concentration of these antibiotics. So here I want to move a bit to a different method. So going away from these spectroscopic methods and look a bit more into atomic force microscopy. And what we can do with atomic force microscopy is quite nicely look at membrane surfaces when they are attached to a flat surface. So this orange circle here that we're looking at is basically an outer membrane vesicle that is flattened out on an atomically flat mica surface. And when we watch the surface of this one with the atomic force microscope, we see that the surface is extremely flat. Now, when we incubate this vesicle on the surface with polymyxins, and then look at the vesicle again with atomic force microscopy, then we see that we get this very strange waffle structure on the vesicle. So there's the same vesicle, you see this really high part here. It's the same in both vesicles, or in both images. And we see that the size of the vesicle slightly increased, and it's completely covered in this waffle structure. And well, this was quite unexpected, and since this was really the first time we could do this with native membranes, we obviously wanted to investigate this a bit further. And what we can see, for example, if we do this with the wild type, so in this case, an MG1655 E. coli strain, then we really get very reproducibly this pattern on the surface, and it's actually quite a regular semi-crystalline pattern, as we can see here from this Fourier transform of the image. Now, when we do the same thing with a polymyxin resistant strain like this MCR1 strain here, then we actually don't see anything happening on the surface. So the surface is completely clean, and this interaction between the polymyxin and the membrane doesn't really seem to happen. Now, we tried to stand with different E. coli strains with a different composition of the lipopolysaccharide. So this long glycolipid on the surface, either with the full version, which actually has this added O antigen on the surface here, then a truncated version, which only has the core sugars of this lipopolysaccharide, and two more even further truncated version, where the last one really just has the lipid A portion of the lipopolysaccharide and none of the sugars on the surface. And we see that basically when we change the composition of this lipopolysaccharide, then also these structures slightly change. So mostly the diameter of these waffle structures that we measure, in case of the wild type, and in case of the wild type with the O antigen here, they are around 10 nanometers. And if we then start decreasing the sugar length on the outside, we also see that these structures seem to be getting smaller, and in particular in this lipid A strain here, we see that they are actually getting a lot smaller. The other thing is that we monitor that the height of these vesicles also decreases quite a lot when we add the polymyxins. So when we look at the vesicle before the incubation with polymyxin, then we get the regular membrane thickness of roughly 6 nanometers. If we incubate them with the polymyxin, then the height of these membranes strongly decreases to around 4 nanometers. And we can actually see in this case the waffle structures in this region is slightly higher than the rest of the membrane, where we don't have the waffle structures. But it's actually quite surprising, because 4 nanometers is really thin for a membrane. And again, when we do this in a concentration dependent manner, we see that this thinning of the membrane and the increase in area of the membrane happens exactly at the minimal inhibitory concentration of polymyxins, and it's actually quite independent of which polymyxin variant we use, whether we use the polymyxin B or the polymyxin E, or also the polymyxin B nonapeptide, which is a slightly different version of the polymyxin where the fatty acid tail is missing. And again, what we see in gray here, this is the control of a detergent, in this case LDAO. The action of the detergent is very different, where we don't see this sharp drop around the minimal inhibitory concentration. So, well, taken together, this is actually quite puzzling, and we are really sure that the emergence of these waffle structures is actually quite important to the mode of action of polymyxins, which previously was like really poorly defined. So this is basically up here the model system, or the model of how polymyxins are supposed to work. And it's a rather obscure mechanism where the polymyxin interacts with the LPS in the outer leaflet of the membrane, potentially displacing divalent ions from the membrane, and then somehow just, well, basically slashing large holes into the membrane. Although the details of this are completely in the dark so far. And what we've now seen for the first time is that as part of this mechanism, somewhere these waffle structures need to form, and actually, well, at least potentially play a really large role to then disrupting the membrane. And again, this is still under investigation. So we've seen the structures, we've seen that these very nicely coincide with the minimal inhibitory concentration of these polymyxins, but we're not entirely sure about the molecular mechanism underlying this action yet. And for this, I'm at the end of the talk. So I hope I could convince you that outer membrane vesicles are actually an excellent system to study outer membranes in isolation using a variety of biophysical methods. Because they, well, for the first time, they really give us direct experimental access to the unaltered outer membrane, which hasn't been possible so far. And we can use them quite nicely, looking at how beta-lactamases work inside of their native environment and how inhibitors get across the membrane to interact with these beta-lactamases. And we can also use them quite nicely to look how membrane-targeting compounds, in particular antimicrobial peptides acting on bacterial outer membranes, interact with these native membranes. And, well, in case you're interested in any of this, in particular, if any of you is working with experimental beta-lactamase inhibitors and is interested in getting them across membranes, we're always open to collaborations, and I invite you to contact me at this email address here. And with that,
thank you, Johannes.
Thanks, and I'm open to questions.
Thank you so much. Interesting, and I love the fact that you're connecting the past with the future. So thank you for that. We've listened to a lot of interesting speakers today. I hope you enjoyed this. The week is full of activities. Like I said, there is an opportunity to join us already tomorrow at the Twitter space. And next week on Wednesday, you will meet other speakers talking about their research. So feel free to join us then. But without further ado, I'm actually going to thank you for joining today. It's been a really interesting session. I hope you enjoyed it and have a great rest of the day. Thank you so much.